Welcome to our educational video on enzymology. By the end of this video, you will be able to learn about enzyme regulation. Enzymes can be regulated by allosteric effectors, covalent modifications, or changes in synthesis or degradation rates. Enzyme regulation is essential for coordinating metabolic processes in organisms. The first method of enzyme regulation is regulation of allosteric enzymes by effector molecules. These effectors bind non-covalently at a site other than the active site that can decrease or increase the activity of the enzyme. The effectors that inhibit enzyme activity are termed negative effectors, whereas those that increase enzyme activity are called positive effectors. Homotropic effectors. When the substrate itself serves as an effector, the effect is said to be homotropic. Most often, an allosteric substrate functions as positive effector, in which the presence of substrate molecule at one site on the enzyme enhances the catalytic properties of the other substrate binding sites. Heterotropic effectors. Your substrate does not act as the effector. Instead, it is other molecules distinct from the substrate that serve as heterotropic effectors. The effector bind to a site on the enzyme separate from the active site and modulate the enzyme's activity, either enhancing or inhibiting it. Most allosteric enzymes are multi-subunit enzymes. They do not follow Michaelis-Menten kinetics. They are key regulators or rate-limiting enzyme and metabolic pathways. Covalent modification. Enzymes regulated by covalent modification is often through addition or removal of functional groups by formation or breaking of covalent bonds. These are some types of covalent modification. Out of these, phosphorylation to phosphorylation reactions are more important. Phosphorylation reactions are catalyzed by a family of enzymes called protein kinases that use ATP as the phosphate donor and add phosphate groups into the enzyme. Dephosphorylation reactions. The phosphate groups are cleaved from phosphorylated enzymes by the action of phosphoprotein phosphatases. Depending on the specific enzyme, the phosphorylated form may be more or less active than the unphosphorylated enzyme. For example, phosphorylation of glycogen phosphorylase increases activity, whereas phosphorylation of glycogen synthase decreases activity. Enzyme synthesis and degradation regulation. Cells regulate enzyme levels by altering synthesis rates, typically increasing for enzymes needed at specific stages or under certain conditions. For examples, insulin increases synthesis of glucose metabolism enzymes in response to high blood glucose. The changes in enzyme levels due to synthesis regulation are slower compared to allosteric or covalent regulation. Enzyme degradation regulation is for short-lived proteins like cyclones. You can get access to YouTube videos along with notes and MCQs through our website, link given in description. If you found this video helpful, please like, share, and subscribe.